Hello, and welcome back to another exciting episode of What is Biology? I'm your host, Mr. Johnny Hopkins, and today we're going to be talking about proteins, the structure in particular. So, there are four different structures of a protein, if you will. There's the primary structure, secondary structure, tertiary structure, and quaternary structure. And for shorthand notation, it's a 1 degree sign, 2 degree sign. So 1 degree sign is primary, 2 degree sign is secondary. Makes sense. Now, the primary structure is what we've been talking about in the previous lecture of the amino acid sequence. So the sequence of residues, where the residue is just a single one of these. So as you've seen before, this is what a protein peptide looks like. It goes from a N terminal to a C terminal, which ends like that. And we have the different amino acid side chains noted by these R's here. Now, and if we remember, this is a single peptide unit. Now, so that's the primary sequence. It's just the sequence of the residues. So, real quick, we're going to get into a few little notation things. There's this thing called a Ramachandran diagram, which in particular, it's mostly just kind of talked about nowadays, but not actually used for a lot of purposes besides just kind of like checking things. And bioinformatics people, they deal with this. Everyone else pretty much just kind of just like, okay, that's cool. So Ramachandran diagram looks like this. It's got this psi and this phi right here. But what the heck are these psi and phi? And what the psi and phi is that for every peptide, like this or like this. So these four atoms pretty much lie in a plane with each other because effectively this double bond with the carbon is kind of shared with this nitrogen as well. And so it just kind of makes this little nice plane. And then you can kind of just compare this plane to the plane of the carbon alpha of the previous following residues. So as I just said, carbon alpha is just the the carbon attached to the amino acid side chain. So that's the alpha carbon, the first carbon of the amino acid side chain is the carbon beta, and then go for it. So it goes alpha, beta, gamma, just follow the Greek alphabet. If you're frat bro or physics nerd, you'll get along just fine. So that's the nomenclature. So what are the psi and phi angles? As I just went down this little tangent, is that we have a psi and a phi angle. So as I said, the plane is essentially what's the angle between this guy and this guy, between this plane and the plane of the amino acid side chain. So the further apart they go, you have a lot of deviation in the psi angle, but not so much in the phi angle. That's effectively kind of what it states, and that has some implicit information. This guy gives rise to the secondary structure. But a little bit of nomenclature first. We're going to arbitrarily define some stuff, which just makes everyone happy when we arbitrarily define some terms that have real no purpose. We've got the difference between a peptide, a polypeptide, and a protein. A peptide is around 20 amino acids. That's what this AA stands for, 20 residues. And so it's usually small and doesn't have a lot of form. It's a wiggly boy, as I commonly say. So they've got a wiggly boy, and a peptide is just like 50 plus amino acids. And then you have a protein, which is a polypeptide that has structure. Or is a wiggly boy, but with very strictly defined wiggliness. That's called an intrinsically disordered protein. So this is extremely arbitrary and not really useful in anything besides just fighting over semantics, which, unless you're Socrates, nobody cares. So now we're going to give rise to the secondary structure. Secondary structure is all about where do the hydrogen bonds go in this protein. So now what is a hydrogen bond? As you might remember from basic chemistry, basic biology, or any sort of pop science stuff, hydrogen bond is just a bond. It's just a bond between hydrogen and a different atom. So, most common example is water. I'm going to draw up here. So, effectively, there's an uneven distribution in this covalent bond between oxygen and hydrogen. They share these electrons, so they're really close together and tightly bound. But the oxygen here is slightly negative, and this hydrogen is slightly positive. So that gives rise to this slightly positive guy getting attracted to the slightly negative oxygen over here. So the same thing happens in the backbone of the proteins. And what happens here is this backbone, this oxygen is slightly negative. This nitrogen is slightly positive. So when you have this oxygen right here attack with that nitrogen, the hydrogen on this one, you can have a hydrogen bond occur. And that helps stabilize the protein itself and gives rise to these funky little fancy secondary structures. These are fancy boys. They wear a bow tie and everything. So you get the alpha helix and the beta sheet. Those are the two primary secondary structures. You've got a few others, but these are the important ones. You've got an alpha helix where essentially the hydrogen bonds just kind of, it's a structure where this long chain wraps around itself such that the amino acid right here, the amino acid above, just kind of hydrogen bond with each other, such that the nitrogen and oxygen above just keep hydrogen bonding. And so what's interesting about this guy is that this guy can form practically anywhere while the protein is being formed. So this guy is very, very quick to form. It's like, you know, just random people showing up to the McRib when it gets announced. This thing happens very, very quickly, instantaneously, if you will, in the grand scheme of things. Then you have the beta sheet. And the beta sheet is essentially just a long line, or a long, like, planar structure, if you will, of this peptide. It bonds with a second part of the peptide, and you've got hydrogen bonds all along this axis. So it's along this axis, which means that it's also along the axis of this peptide backbone bond, rather than using that as the normal, which. So, and it points from N terminus to C terminus. And beta sheet, this guy doesn't form with, doesn't have a hydrogen bond with the, between the current residue and the residue that's far away. This can have a hydrogen bond with anywhere along the protein chain. So this guy takes a long time to form because it can take a long time for one thing to get close to another. It's just kind of randomly has to interact and get there, and then it forms these tight hydrogen bonds that help stabilize the structure and give it this form. And so when we go from sequence to this part of the hydrogen bonding, then we can go to the 3D structure, because this interaction with the local environment causes a 3D structure to form, because we're talking about proteins here and not wiggly boys called peptides. So tertiary structure is just the 3D structure. What are the grooves, and what are the grooves of it? Is it smooth? Is it flat? Is it bumpy? Does it look like the topological map of Kansas or of Colorado? And that is practically all tertiary structure. And one interesting thing is that the tertiary structure also takes into account this thing called disulfide bonds. If you remember, there are cysteine residues in proteins, and these cysteine residues have a sulfur on them. And as I discussed previously, these sulfurs can be reduced and actually bond with each other, such that they help, they bridge the gap, and kind of like hydrogen bonds, they help stabilize the molecule itself. that was a big development into the studies of Anfinsen, if you remember that guy with his fancy sequence-defined structure. And so, additionally, one thing that is interesting about the 3D structure is we have these things called motifs and domains. A motif, there are 3 ones outlined here. We have a coiled coil, where we have 2 alpha helices, where they can just kind of go right by each other. And typically the region of where they interact and overlap, so right where the X is, those are filled with hydrophobic residues that kind of just interlock, and that helps keep them safe from the water around it. Then we have basic helix loop helix, BHLH, basic helix loop helix. That is a helix, then you got a little loop, and then you got a helix, and that is a very common structure that happens in DNA binding. And then finally you have a zinc finger, where you have an alpha helix and then 2 beta strands, and they just form some hydrogen bonds and different interactions with the amino acid side chains that cause it to bind zinc very well, and those zinc fingers can be used for different functions in the cell. Then finally we have this thing called a domain. Now what a domain is, we're coming back to some arbitrarily defined definitions. Socrates would have a field day with biology. So a domain is just a structurally and functionally distinct section of a protein. How do you define a structurally and distinct function of a protein? It's kind of arbitrary. But usually there's some genetics that you can kind of look at and be like, this region, it shows up in 3 different genes, and it's always the same thing, so you're just kind of like, it's probably a domain. And they look at it and think, it's a domain. But in general, it's kind of easy to identify, but quantitatively you can't really define it. So you have essentially just these distinct regions that can operate independently. And there are a lot of proteins that have like 15 plus domains that all interact independently and help form this entire complete structure in this just single protein. And now there's a quaternary structure. So we're just talking about these domains that are functionally independent. But what about if we had different proteins just kind of combined together? Because like what I ask myself every morning when I wake up is, what if I'm not good enough? And this answers that question. We can't just depend on ourselves sometimes in society. We have to depend on all those around us to help get this, to help achieve the single functions or a single goal. We have the quaternary structure, which essentially multiple peptide chains, or protomers, if you will, join together to form a protein complex. So you can have multiple different proteins interact to form something like this. A hexameric structure is this guy, which is found in ATP, AAA plus ATPases that can help degrade proteins. And as well as we've got a hemoglobin, which is formed of 2 protomers, or different structures that are homodimers that can assemble on the membrane of the cell and induce cell signaling of something, along with a bunch of other different quaternary structures possible. Then finally, now we know, okay, we got a structure of a protein. That's cool. But like, how does it get from, kind of being in a ribosome and just kind of flopping out and being a wiggly boy, and then coming back and going to this compact, beautiful 3D structure that just magically uses ATP and makes me just think about applesauce all the time. And that's what happens with this. I'm going to tell you why you think about applesauce all the time. Folding. This protein folding is studied by a lot of different groups, and it was pioneered by this guy named Dr. Ken Dill, who's currently at Stony Brook University. So what he looked at is this thing called the folding landscape, where effectively you have this random coordinate, a reaction coordinate, if you will, shown here. That's this x-axis, and this y-axis is shown as the energy. U, potential energy, or delta G, gives free energy. Change in gives free energy. So what we have here is effectively this side. At these ends, we have a wiggly boy at some weird conformation. And then we have here the native structure, also usually noted by an N. And this is the unfolded structure, or U. And then along this path of folding, we get these little minima right here that are intermediates, or I. Now, what is important to note is essentially like filling up sand. The sand wants to go to the lowest point, or the lowest energy. The lowest energy conformation means the most probable or most likely conformation. And that protein landscape goes towards that because it's the most energetically stable. It's akin to this fancy little 3D structure. Now, what drives this? Why do we get this fancy funnel thing? And that is electromagnetic forces. Electromagnetic forces define everything of protein folding in a classical sense. You have, and then we can just subdivide it into 4 different types of electromagnetic forces that help define this. You have coulombic interactions, hydrogen bonding, van der Waals forces, hydrophobicity. The 2 that are the most important, arguably, are hydrogen bonding and hydrophobicity, with a lot of studies arguing that either hydrogen bonding is the most important, or hydrophobicity with hydrophobic collapse being the most important. But arguably they're pretty much the same. So what is a coulombic interaction? You have a plus Q and a minus Q. And what that just pretty much means is that if you have a positive charge and a negative charge, they want to be close together. If you have a negative charge and negative, they don't want to be close to each other. Like 2 brothers sitting in the back seat of a car. They don't want to be next to each other, so they're constantly fighting and fighting and fighting until they have to separate them. Same thing with positive charges. They don't want to be together. So that helps form and make things a little bit stable or repels things away to help an alternate conformation exist. Now in hydrogen bonding, as we just previously discussed here, hydrogen bonding is essentially like this coulombic interaction, pretty close, but it's not anywhere near as positive and negative in charge, and it's driven by hydrogens rather than actual positive charges on heavy atoms such as oxygen and nitrogen. So that's hydrogen bonding, and that forms those secondary structures. Now we have van der Waals forces. Effectively, you can't get inside an atom. They repel each other just from getting close enough together. It's all about having your safe space, your personal space. That's effectively van der Waals. You just can't go inside. You can't have another carbon and another carbon get close enough to each other. Otherwise, you have an atom bomb, and that's not good. So they can't get close enough together because then that'll cause fusion. Bad for literally everything in everything organic. Then we have hydrophobicity. Hydrophobicity is effectively certain residues don't like to interact with water. In general, if it's a carbon or a lot of carbons in a molecule that doesn't want to deal with water. And what typically is observed is, as previously stated, we have a protein that kind of looks like this, and we just take a cross section in the middle. This center is hydrophilic. This center is hydrophobic. This outside is hydrophilic because the outside is going to be surrounded by water in the cytosol of the cell. Because the inside of the cell is the cytosol or cytoplasm, which is primarily made up of water and a bunch of other proteins and other stuff just floating around in a weird environment. And this inside is hydrophobic, so it stays away from the water, and the outside preferentially works with water so that it can move about, so it can go and do its job. So these things drive protein folding. But as I just said, in the cytosol, there are a bunch of other proteins. And what if some of these proteins helped fold other proteins? That would be a novel idea. And actually some of them do. And in the cell, you have these guys called chaperones. These guys help take them to the apple orchard. These guys help make them so they can go to the apple orchard, so they don't have to stay home and eat a peanut butter sandwich out of a sack lunch wishing they were at the apple orchard. Chaperones, proteins that fold other proteins. There are, in general, there are 3 of them that are highly talked about and discussed and studied, but there are thousands of different types of chaperones. There are hundreds of different types of chaperones. We'll talk about 3 real quick. Trigger factor. So the ribosome is this giant RNA protein complex that makes proteins. And it's shown here as these 2 little ovals. And then you have this blue nascent peptide chain. So this guy, it's exiting as a wiggly boy. And then it attaches to this guy called trigger factor, which is called a dragon-shaped protein. Even though it doesn't look like a dragon, sadly. And it binds to that and it helps fold this peptide as it exits the ribosome. Nice. You have HSP40 and HSP70, which also do similar things after the peptide chain exits the ribosome. They're also something called heat shock proteins, which help stabilize the protein in unfortunate hot conditions, as well as something called a chaperonin. So, as I said, this cell has a bunch of proteins floating around in it. And what if another protein kind of just randomly hits this nascent peptide chain or this slightly unfolded peptide? That's not going to be good, because that might change this energy landscape to be something else and then have a different negative state, so it has a different function, so it hurts the cell. That's bad! So, chaperonins exist. So if the protein doesn't fold by trigger factor in these guys, it can go to this guy called a chaperonin, which is made up of 2 proteins in a different coordinated structure, GRO-EL and GRO-ES. So GRO-EL is essentially just a cylinder with a hollow center, and then you take a wiggly boy, you throw it in there, and then it folds, and it folds such that it no longer is confined and around all these other proteins that are bad influences telling it to do drugs. No! GRO-EL is going to help it avoid peer pressure and come out into a strong, independent protein with hydrophobic residues on the inside and hydrophilic residues on the outside. GRO-EL is a good role model, and we should all be a lot more like him. GRO-ES is the lid on top of GRO-EL that essentially helps it stay as well, such that nobody else comes in and tries to influence it while it's teaching it good things. So, that is the general overview of protein structure. So, protein has a primary structure, the sequence, a secondary structure, which is just the network of hydrogen bonds that give rise to a 3D structure. And this 3D structure has different little motifs of how these secondary structures interact with each other in a 3D location. Then that can give us a function, but sometimes multiple proteins need to combine together in order to form a protein complex that has actual functionality. And the ways that this protein can get into these folded structures is by folding along an energy landscape and finding the lowest energy structure, which gives it the proper function. This energy landscape is formed by electromagnetic forces, which there are 4 different types of electromagnetic forces that are primarily involved: Coulombic interactions, hydrogen bonding, van der Waals forces, and hydrophobicity. And sometimes it's not enough for a protein to just kind of escape as a wiggly boy and fold on its own, because the cell's a very crowded environment, like Times Square. Hate Times Square. It's too crowded. So instead of staying in Times Square, it can be helped by chaperones that help fold it into its native structure. The chaperones include trigger factor, HSP40, HSP70, and chaperonins such as GRO-EL and GRO-ES. So that's all I wanted to discuss today about protein structure. Next time we'll be talking about protein binding, as well as protein degradation, and finally wrap it up with some experimental methods into studying these proteins. I'm Mr. Johnny Hopkins, and this has been What is a Biology? Thank you.